And in certain creatures like lobsters, they seem to have an unlimited supply of this enzyme, and so lobsters can theoretically just live forever. You need DNA to make enzymes. You need enzymes to make DNA. How do you get them both at the same time? You can't. It's like asking the question, which came first, the chicken or the egg? Redeem Academy. Hey, what is up, Science Squirrels? Johnny here with Redeem Academy. I'm here to help you get the most out of your education while giving God all the glory he deserves. We're in the middle of a DNA unit. Lesson 4 today, DNA replication. I'm going to be telling you about 3 things today. How to properly write a complementary DNA strand, how tiny machines inside your nucleus make perfect copies of your DNA, and why, considering DNA replication, intelligent design is the best explanation for life. Big question today, why is intelligent design the best explanation for the existence of the DNA replication system? We will get there. All right, I wanted to start out today with the term replication. Do you know what replication means? Well, here is a video from the internet that I found that involves replication. Any idea what they're making replicates of in this video?
Rolling in the Benjamins.
You know, after watching this video, I thought to myself, you know what? I need to get one of these for my house. It's kind of big, but I think it would probably fit in my garage. And it could really come in handy for some of the things I've been wanting to get. I mean, it only makes one thing, but I think I could use that one thing to get some other things. It's really good at replicating $100 bills. Oh man, that's a lot of $100 bills. I wonder how many $100 bills are in one stack like that of $100 bills. And then it puts a nice little wrap on it, like kind of like a birthday present, you know, comes in its own package. Now these people that work there and just move stacks of $100 bills around every day, that's an interesting job. I wonder if like once every year somebody snaps and just grabs a stack of $100 bills and just takes off. Anyway, Christmas presents, Mr. Olkers would love a $100 bill replicator machine. Amazon? So anyway, the word replication means to make copies. Whenever cells are going to divide, you have to have a complete copy of the DNA, and the chromosomes rip apart, and your chromosomes are your DNA. So I've got some fast facts for you about DNA replication, which is the process of making a perfect copy of your DNA so your cell can divide and have all the instructions for how to be a cell. All right, here we go. Fast facts on replication. So first off, replication has to happen during interphase before the cell has begun mitosis. So remember, interphase is that first part of the cell's life where it's in use and it hasn't started the process of mitosis yet. 9+ enzymes are involved in DNA replication. Now you're about to see this, but enzymes are tiny little machines that come and grab on and do things to the DNA molecule to put it in the right position, to put all the right pieces in place. There's 9 different enzymes involved in humans for doing this. They're like tiny little machines that God designed to actually make the right copies of DNA, because you got to get this thing totally perfect. And remember, it's jammed inside the nucleus of a cell. It's like as much information as 100 encyclopedias stuffed inside of a cell, and you got to copy every single letter correctly. This is happening at any given moment in about 1% of the cells in an adult's body. Probably more in a teenager. That equates to 300 billion cells. Now here's something even more mind-blowing. Every time the body makes a copy of DNA, a little bit of the tip breaks off, and this is one way we can track aging. But there's a special enzyme for rebuilding the tip. The tip is called a telomere. And there's an enzyme called telomerase, telomerase, that can actually rebuild the tips. And in certain creatures like lobsters, they seem to have an unlimited supply of this enzyme, and so lobsters can theoretically just live forever. But humans, their telomeres break off, and there's kind of a limit to how long their cells can live because of this. It's called the haystack limit. Scientists think if they could increase the activity of that enzyme telomerase, they could lengthen human lifespans. And this is one idea Christians have about like maybe people lived longer in the Old Testament because it seems like God sort of reduced lifespans in the Old Testament around the time of Abraham and Noah. Maybe part of what God did was he reduced the activity of telomerase, and if you could kick that activity of telomerase back up, maybe you could make humans live a little longer. But you might find yourself fighting against God on that. All right, last one. DNA replication is a chicken and egg problem for people who believe in evolution. In order to replicate DNA, you need certain proteins. But in order to make proteins, you need DNA. So which one came first? You need God to make them both at the same time. It's like asking the question, which came first, the chicken or the egg? I think the chicken came first because God spoke and it came into existence. All right, so here's where we're going to be starting today. This is called DNA replication. So this is a great first video to help you begin to understand what DNA replication looks like. It's very basic. And they're showing you here that DNA in the middle is connected by hydrogen bonds. And those glowing lines are hydrogen bonds, and as DNA helicase comes through, it sort of looks like it's stretching out the hydrogen bonds. They sort of look like rubber bands being broken. DNA helicase is unzipping the helix. And so his whole job, he's an enzyme, and he comes through and he unzips the helix and opens up what's called a replication fork. And boop, boop, boop. It's kind of satisfying to look at. I love how they stretch them out. And certain nucleotides are attached by 2 hydrogen bonds, and other nucleotides are attached by 3 hydrogen bonds. And now we have this big yellow molecule. Now this is not what the molecules really look like. It just has simple little pieces. But this other molecule is called DNA polymerase. And DNA polymerase comes in and attaches new nucleotides. And notice how the triangular shaped ones attach to triangular shaped ones and the smooth ones attach to the smooth rounded ones. And so DNA polymerase comes and puts the nucleotides in place, A's with T's, C's with G's. There we go. We're zooming in. The C's and G's are kind of rounded. The A's and T's are triangular. You get 3 hydrogen bonds with C's and G's. You get 2 hydrogen bonds with A's and T's. And they are chemically bonded. But the weakest bond is in the middle, so it's easy to unzip DNA. And that is the magic of how it works. You get a complementary strand, which is called the daughter strand. You get a template strand, which is called the parent strand. And so you unzip the original DNA, and you make 2 pieces of DNA from 1. Now the reason this is a brilliant design is it's really hard to mess it up. Okay, so here's half the DNA right here that just got unzipped. And for every T, the enzymes, the little machines know, oh, okay, this one becomes an A. And I'm actually going to flip this over. So for this T, we put in an A. For this A, we put in a T. And I'm going to go all the way down. So God made a brilliant design there where 2 pieces of information split apart, and they each just replicate on the complementary side and become a full DNA strand again. It works perfectly, and there's already the code right there, so you know what to put alongside it. That's part of the reason. In theory, it's hard to screw that up. But as I just went down that, I don't know if I made a mistake, because it's easy to make mistakes. But for chemicals, it's not about reading letters like with your eyes. It's about the feeling of molecules. And so these chemicals go down and they feel the molecules. And if it doesn't feel right, it pops out and then puts the right one in. And God designed these molecules to be extremely efficient and accurate in what they do. And so there are very few mistakes. Let's go look it up. All right, this is ChatGPT. He's basically my best friend. He's not really a he, but I call him he. Okay, I just asked, how often does a nucleotide get put into the wrong place when a human cell is replicating DNA? Now, there's 6 billion letters in one person's DNA in one cell. And this is cool. I mean, he says, wrong nucleotide gets added sometimes, but those mistakes get fixed. So God not only built molecules that do this, but then he built molecules that come behind it and proofread it. That's incredible. So raw insertion mistakes, 1 out of 100,000. That's a lot when you're doing 1 billion letters. After proofreading, 1 out of 1 billion. I'm going to say, do those 6 nucleotide mutations for every new cell cause a person to age? I think maybe part of that is aging. Okay, and in summary, it doesn't usually hit coding DNA, so it's not going to affect proteins. And that's another good point. A lot of the cells that get made, that's the final cell. They're not stem cells that are going to become more cells. Amazing, amazing system built into us, working all the time. It's like riding a unicycle while copying an encyclopedia during an earthquake. Now I'm going to show you a few of the specifics in our notes. Okay, so first off, what's with me doing it upside down right here? Okay, remember on the first day, drawn a little better, these nucleotides are upside down. And we have a word for that. DNA is anti-parallel. It's like a two-way street. Okay, so DNA is anti-parallel. So I brought some toys along. Here is a sweet race car, and here is the police.
It's the popo. Be cool.
And they drive this way, and they drive this way. So the cars go in both directions. The race car goes this way, the police go that way. It's a two-way street. And that's how DNA operates. The cars go this way, the cars go that way. It's a two-way street. So that brings us to this giant thing. This spot right here is called a replication fork. So scientists have actually been able to take DNA out of cells and get them to open up and start replicating. And so each one of these bubbles is a different spot on the DNA strand where, you got two there, where the DNA has opened up and started to replicate itself. So it doesn't just start at one end and then wait for it to go all the way to the other end. The DNA will open up all these little replication bubbles. There's so many of them. And it'll start replicating itself. And so just one little end, I'm going to put a box around this one. This is a replication fork, where the DNA has forked in the road and is starting to replicate itself. And through a series of lots of experiments in the 60s, 70s, 80s, and 90s, scientists were able to figure out all the things going on at the replication fork. Now, we already saw two enzymes. There's one called helicase in the first video that was orange. Helicase unzips the DNA. And its job is just to unzip the DNA right here. And so this represents the original DNA that's been unzipped. And so I colored the original DNA green. And then the other guy that in the video was yellow was called DNA polymerase. I'm going to color these guys yellow. And his name's DNA polymerase. So remember, a polymer is a chain. And so polymerase, anything that ends in ase is an enzyme. And an enzyme is a little machine in the cell that does work, made out of protein. And so polymerase is a protein that builds polymers. Helicase is an enzyme that unzips the helix. All right, so here's where my cars come back in. There's my car driving that way. Here's my police car driving that way. So it turns out DNA polymerase can only drive in one direction. So the DNA molecule on this end that's being built is called 5', and on this end that's being built, like right there, is 3'. And that's like saying the far away end and the close-up end. And so this molecule that represents polymerase, he is driving 5' to 3', and that's the only direction he can drive. And then this guy, same thing, he can only drive 5' to 3'. And so what he does is he reads the old DNA strand, the green one, and then he lays down the new nucleotides for the new one. So maybe this part's even hard for you to understand. All these letters, they're on the DNA. I've just zoomed out so far we can't see the letters anymore. And so what this guy's doing, DNA polymerase, is he's reading the letters of the green strand, and he's driving forward and he's laying down the next letter every time. So just like we wrote A, T, G, G, A, C, G, DNA polymerase's job is to drive down the green one and then to lay down the correct letter. So if he comes across a G, he puts down a C. If he comes across a C, he puts down a G. If he comes across a T, he puts down an A. If he comes across an A, he puts down a T. And he just keeps driving and laying down letters over and over and over again. And the only direction he can drive is from the 5' end toward the 3' end. Let's even write the word end down here. 3' end, 5' end. Okay? So, because helicase is unzipping the DNA, right here, and he's continuing to drive himself. He's continuing to fly like a spaceship into the DNA molecule and unzip it. The DNA just keeps unzipping in this direction, and DNA polymerase, he just keeps driving in this direction, and so it's like a permanent unzipping motion all the way down. That's called the leading strand. He's leading the way. This helicase just unzips and moves forward, and DNA polymerase can just move forward in this direction. Yellow represents the enzyme DNA polymerase, and he can just move forward and unzip and just keep going and going and going. But on this end, the car has to go and drive and then fall off, and then go and drive and fall off. And then more opens up, and he goes and he drives and he falls off. And he has to keep driving and falling off just because helicase is moving that way. And so because of that, this side is called the lagging strand. He's lagging behind. He's going and falling off and coming back and going behind and falling back. And he keeps going and he's catching up, but he's slowly catching up and lagging behind in his progress. So it's called the lagging strand. Now these little problems have to do with the shape of molecules and what molecules can and can't do based on their shape and the shape of other molecules they're copying with and working alongside. And these are like engineering problems. And so there are people called molecular biologists, and they study molecules and they study biology, and they've worked these things out over the last decades. This was known before I even went to college. And now we have little animations that show us how this works. So now I'm going to show you a more advanced animation of this same idea. Okay, so here we go. We see DNA, it's unwinding because we're learning about DNA replication. And the narrator of the video is probably telling us that T's go with A's and C's go with G's, the stuff we already know. But now we're going to see the enzymes come in, and they're telling us, hey, there's a 5' and a 3' end of DNA, and DNA is anti-parallel, so they go in opposite directions. Now they show helicase as a big bad enzyme, and that's probably closer to the truth. Some of the cartoon animations show it as like a really simple little wedge, and others show it probably more like it really is a big funky looking protein. And here comes another enzyme. I told you at the beginning there's 9 enzymes, 9 different molecular machines involved in replicating DNA and getting it right. So this other one comes in and he just starts a little primer, and then that falls off, and then that primer is actually going to be removed eventually. And you can learn much more advanced versions of this in biology in high school and college, higher end level classes. But then we start going 5' to 3', and that big chunk of brown stuff there is DNA polymerase. And he is part of the leading strand. But now we're going to zoom in on the lagging strand and look at how it happens on the lagging strand. And there's a famous scientist who named the little fragments. And we got to start off these little primers again, and you don't really need to know about that. But then DNA polymerase comes, he grabs on, he rides for a little while, and then he has to fall off, and he has to go back and start all over. And I try to skip over even the part about the primers, but DNA polymerase goes and jumps on and makes a copy again. And then there's some other things that come in and remove some things and do some other stuff. It's very complicated. It is not simple. And that just kind of goes to show, like, life doesn't just come about accidentally in a puddle if you strike it with a lightning bolt. Like, everything going on in life, so complicated, so complex. Let me just move to this point really quick. I mean, I'll spend a little more time on this in the big question, but if you come across certain features like these rocks right here, you look at that and you go, yeah, maybe time and erosion produced those rocks. That's a product of nature. But then there's a certain amount of complexity and specificity to a thing where you come across it and you go, nature didn't build that. That was the act of an intelligent agent. And when Darwin was around and he was coming up with evolution, he didn't know anything about cells. And he'd look at cells and he'd go, well, there's some kind of tiny microscopic thing there, but maybe it just sort of comes together through like simple means. But here we are 200 years after Darwin, or whatever it is, 150 years after Darwin, and we're looking at cells on the inside and we see nothing but amazing complexity in machines, rebuilding perfect codes inside of us. It doesn't look like some rocks that were shaped by water. It looks like some statues that were built by an intelligent mind. All right, so as our little car drives down the DNA molecule, pink gets laid down and builds the new piece of DNA. So I'm going to color it pink. Okay, same thing on this time, except this guy's driving down and he's laying down the pink in little fragments. Okay, and they have already formed down here. So I'm going to color these guys pink. Now originally we started with one green strand of DNA, double-stranded, and that was called the parent. And so it's called the parent strand that gets copied from, and then the new strands that get made are called daughter strands. All right, got a bunch of words to fill out here. This whole process is called replication. The stuff that we need to copy is DNA. The enzyme that does the unzipping is called helicase. The enzyme that reads the DNA and puts down the correct nucleotide on the growing chain, that's called DNA polymerase. The old DNA strand in the new DNA molecule. So we have two new DNA molecules that are being made, this one and that one. The old strand is the green one. It's called the parent strand. Okay? The new DNA strand, so look at both of these. They both have a green and a pink in them now. The green was original. The pink has been rebuilt. The pink one is called the daughter strand. The DNA strand where the DNA polymerase can copy non-stop. So look back at our picture. DNA polymerase is always, let's add this arrow, he's always driving this direction. Okay, and remember, there's a 5' end and there's a 3' end. He's always going in the 3' direction, so he always drives 3' to 5'. Oh, but the question is asking which one is doing that. It's the leading strand. And the DNA strand where DNA polymerase has to fall off, go backwards, get back on, and start copying it again and again. Okay? He's doing that over and over and over again in what is called the lagging strand. All right? As I just mentioned, the only direction DNA polymerase can drive is 3' to 5'. The place where DNA replication happens is the nucleus of every cell. And the bonds between nucleotides that DNA helicase unzips, this is the first thing I should have written down. Those are called hydrogen bonds. Hey, sorry about that. I kind of forgot to write all those words down as we went. Back to the big question, why is intelligent design the best explanation for the existence of the DNA replication system? I've kind of already told you. You need DNA to make enzymes. You need enzymes to make DNA. How do you get them both at the same time? You can't. There are scientists who are called materialists. They only want to believe there are material things. They don't want to believe in a supernatural world. They don't want to believe that we have a soul. They don't want to believe there's a God. And so they are fully committed to coming up with a natural explanation for how one cell could just suddenly come into existence. That first cell has to have DNA or something like DNA, and you got to have protein, or you got to have RNA, and you got to have protein. And none of those things make themselves. It doesn't work. You know, I could see why, like 2,000 years ago, if somebody came up with an explanation that these rocks were made by giants and shaped like this, some people would be like, I don't think so. Other people would be like, you're onto something. And it could be controversial, like, yeah, I don't know, I think maybe just, you know, or someone would be like, no, God supernaturally shaped them just like that. And you might win some people over and lose some people on that one, because it kind of looks like something maybe just erosion could do. But if you come across something like this, it is complex and it's made to specifically look like something, and there's nowhere else on the planet where anything looks like this. And just like I ran through this before, something like this we can attribute to nature. Something like this we have to attribute to intelligence. In the same way, when we look inside of cells, for the last 75 years, scientists have been looking inside of cells and they've been finding design. They've been finding complexity and specificity. And the only known place where those sort of things come from is a mind. Where is there a mind powerful enough to design things that complex and sophisticated? It'd have to be someone like God. And so there is a movement in science that is growing momentum. It's called the intelligent design movement, and it's exciting to watch. They're still the underdogs, but they're winning people over. And I think they've got, by far, the best explanation for the origin of cells. Hey, I hope this was an interesting lesson for you. It's pretty nerdy stuff. Nerds rule the world, though. Have a great day. Peace out, brown trouts.